Algae are diverse photosynthetic organisms that range from unicellular microorganisms to multicellular forms like seaweeds and are found primarily in aquatic environments. They play a crucial role in ecosystems by providing oxygen and forming the base of aquatic food chains. There are a couple different types of algae. We have green algae, which are mostly found in freshwater environments and are similar to land plants in terms of their chlorophyll content. Red algae are predominantly marine, and they're characterized by the presence of phycoerythrin, which gives them their red color. Brown algae are mostly marine as well, and include large and complex seaweeds like kelps, and are brown because of their presence of something called fucoxanthin. Furthermore, we have diatoms, which are unicellular and mostly found in oceans, and they have a unique silica-based cell wall. Dinoflagellates are mostly marine plankton and can be bioluminescent, and some species are known to cause harmful algal blooms. Blue-green algae are technically bacteria, but they perform photosynthesis, so are often grouped with algae because of their photosynthesis. Moving on to reproduction, they can be grouped into three different types: asexually reproducing, sexually reproducing, or both. Starting off with asexual reproduction, the diatoms mostly reproduce through binary fission. Some sexual reproduction occurs less frequently, but the majority of diatoms will undergo binary fission to reproduce. We have green algae as well, which primarily reproduce asexually through binary fission as well. So they're also classified in this region. And then moving on to sexual reproduction, we have red algae, which have complex lifestyles with alterations of generations, although some reproduce asexually through spores. And moving on to both, we have green algae, which is asexually through binary fission or fragmentation, and sexual reproduction involves the formation of gametes. Brown algae also have both, which is asexual reproduction through spores, while sexual reproduction involves gametes. Dinoflagellates are asexually through binary fission and sexually through the fusion of gametes. And all the ones that go through binary fission is a process where each daughter cell inherits one half of the other's silica shell, and it goes from there. So there isn't an exchange between two cells, it's just by itself. Moving on to the medical importance, we have a couple different things that we can learn from algae, like their nutritional benefits, because they're typically rich in proteins, vitamins, and essential fatty acids that are used in dietary supplements for their health benefits. Pharmaceutical applications, some algae products are bioactive, and which means they can be used in drug development, such as anti-cancer, antiviral, antibacterial agents. It's also some more like learning about toxins and disease. So like certain algae, especially some dinoflagellates can produce toxins leading to harmful blooms that pose health risks to humans and animals through contaminated water or seafood consumption. And there's also research and clinical studies like studying their cell biology, the genetics, photosynthesis of algae, and furthering our understanding of what it all means for all of us.
Hi there, my name is Annika Olson, and I'm going to be talking about different types of protozoa today. So protozoa is defined as heterotrophic, unicellular, eukaryotic organisms that are not fungi. So since it's a very diverse group, they are more so defined by what they are not rather than what they are. They can also be classified by their type of movement. And they are considered pleomorphic, as for they lack the cell wall. So the first type of protozoa would be amoeba, and they move by the extensions and retractions of their cytoplasm, and they reproduce through meiotic binary fission, which is like asexual mitosis followed by binary fission. They're typically found in the intestines if ingested, but are commensal, meaning they live in a host without causing harm most of the time. And an example of a disease of this would be amoeba biasis. And then ampicomplexans reproduce through sexual and asexual reproduction. Medically speaking, they are intracellular parasites, which means that they require a host cell to replicate. And the most known disease of this would be malaria, which we've all heard with the mosquitoes. And then we have hemoflagellates, which move, as it says in the name, by phalange. And again, they reproduce through meiotic binary fission and asexual reproduction. They have chenoplasts, which is helpful for treating diseases by this type of protozoa. And an example of this would be the Chagas disease. And then lastly, we have the looming dwelling phalangites, which again move by phalange and reproduce through meiotic binary fission as well as asexual reproduction. And they are found in the intestinal tract or genital tract, and the disease most known associated with these is Giardia, which affects your stomach and GI tract.
Slime molds act and look like fungi, but at a cellular and molecular level, they are more closely related to entomoebal species. There are two types of slime molds. There's cellular slime molds and plasmoidal slime molds. Cellular slime molds live as single-celled amoeba that ingest bacteria on decaying vegetations. When nutrients are scarce, they aggregate into a moving mass called a slug. Some cells form a fruiting body while others become spores. They play a crucial role in soil ecosystems, regulating bacterial population and organic matter decomposition. They're found in various environments, including forest soils, grasslands, and caves with species diversity influenced by geographic and ecological factors. Plasmoidal slime molds are large, multinucleated superamoeba that can reach 0.5 meters in diameter. Easily visible to their large size and bright color, they are formed when haploid spores germinate and fuse into a diploid cell, which undergoes repeated nuclear division, creating a multinucleated plasmoidum. The plasmoidum moves over decaying wood and leaves, ingesting organic debris and microorganisms. They exhibit cytoplasmic streaming and can recognize the microfilaments for movement, with species distribution affected by environmental factors such as moisture and elevation. When food or water is scarce, it forms spore-bearing fruiting bodies, resisting the cycle. And that's it on slime molds.
So I will be talking about our water molds, which are also known as oomocytes. They are fungus-like organisms that belong to the phylum Mycota. It contains two characteristics. We have our cell walls, which these water molds tend to have cell walls that contain cellulose. And then for our flagella, our multispores, also known as zoospores, contain two flagella. These organisms work well in aquatic environments, such as parasites and moist cells. Also, these organisms play an essential role in various ecosystems. And some examples, we have our saprolegnia, which is an important decomposer for various ecosystems. And then we have our Phytophthora, which contains species which causes plant diseases. And yeah.
Hi, my name is Reagan, and I will be talking about viruses in human tumors. A tumor is an abnormal growth of tissue resulting from a malfunction in a normally high regulated process of cell growth. Some tumors are benign, meaning they do not invade nearby normal tissue, and others are cancerous or malignant, meaning they have the potential to grow and become cancerous in the body. Control of cell growth and division involves two sets of genes. those that stimulate growth, called proto-oncogenes, and those that inhibit growth, called tumor suppressor genes. Mutations that either increase the expression of proto-oncogenes or decrease the expression of tumor suppressor genes are the most common cause of abnormal and or uncontrolled cell growth. An oncogene is a proto-oncogene that has been changed in such a way that it promotes uncontrolled growth, leading to tumor formation. Most tumors are caused by mutations in host genes that regulate cell growth, but some are caused by viruses. In contrast to the cancer-causing viruses, other viruses offer potential for treating cancers. Viruses that can cause cancer in humans are called oncoviruses or oncogenic viruses. The cancer may arise as a direct result of viral infection, such as when a viral gene functions as an oncogene, or when the viral genome inserts into the host cell chromosome in such a way that a proto-oncogene is converted to an oncogene. Alternatively, the cancer may arise indirectly as a result of DNA damage that occurs during long-term inflammation associated with the chronic virus infections. Most virus-induced tumors are associated with certain DNA viruses, although some RNA viruses are also oncogenic. Cancers caused by the viruses do not develop immediately, but instead become apparent between 15 and 40 years after the infection. Some virus-associated cancers are preventable with modern vaccinations. In contrast to oncogenic viruses, oncolytic viruses specifically target and kill cancer cells, a characteristic that makes them potentially useful in cancer treatment. Some oncolytic viruses destroy the cancer cells directly by multiplying them within them and inducing lysis. Others act indirectly by stimulating the host's cancer-fighting immune cells. Although naturally occurring oncolytic viruses were described decades ago, scientists are now genetically engineering other well-characterized viruses to develop oncolytic versions for therapy.
Cultivating and quantifying bacteriophages. Bacteriophages are viruses that infect and replicate only in bacterial cells. Cultivating bacteriophages. Plaque assays are used to find the plaque of phage particles in samples such as sewer water and soil. To do an assay, a melted, cool, soft agar medium is inoculated with both a bacterial host and phage containing specimen. The mixture is then poured over the surface of the plate to create a top layer over it. Some simple steps to cultivate are grow the host bacteria, infect the bacteria with the phage, and isolate the phages from the plaques. Some factors to consider are that the selected temperature and strain of chosen host can affect the infectivity of the phages. The biology of type of phage, considering its burst size, absorption rate, etc., can also play a factor in infection process. For quantifying bacteriophages, a sample contains enough viral particles to be seen through a microscope. Quantifying can be used to determine number. Virus titer is the concentration of infectious viral particles in a sample. Some different methods of quantifying bacteriophages used are the quantile assay, which is a method that uses several dilutions of a virus culture administered to appropriate hosts to determine virus titer. And then the hemagglutination assay, which is a test that relies on clumping of red blood cells to determine the relative concentration of viral particles.
So when growing animal viruses for research, we need to use different types of cell cultures to help them grow. So the one method we use is called homogeneous culture. And this is where the cells flow in liquid. And this makes it able to control contamination for the cells. Another cell culture we use is the microcarrier, and that is where cells stick to small beads to allow them to grow and attach and multiply. And the last one we use is we have these cells called diploid cells, and these are special cells that they're similar to our human cells, but they're great for making viruses grow. And all these techniques help scientists grow certain viruses and use and figure out what kind of techniques or what kind of medicines they need to provide to kill these viruses. So when scientists need to measure or count animal viruses, there's a couple different methods that we use to do that. So the first one is the direct microscopic counting. This is where they look at viruses right underneath the bacteria and count them directly. Another method is called culture counting, which counts the particles by measuring changes in electrical resistance, which counts the cells based on the scatter of light. There's also plate counting, which I feel like is the most common one. This is where scientists grow bacteria on the plate and they count how many colonies form on those plates. And each colony comes from one single bacteria. And all these methods help to tell scientists and measure viruses and understand their behavior.
The Christian worldview behind microbiology. The study of microbes reveals the intricate creation and work that the Lord has done. Most scientists research to not only learn about the microbe itself, but to also learn how to take care of the environment in a way that glorifies God, considering He made all things. Matthew chapter 25, verses 35 to 36 says, "For I was hungry, and you gave me something to eat. I was thirsty, you gave me something to drink. I was a stranger, and you invited me in. I needed clothes, and you clothed me. I was sick, and you looked after me. I was in prison, and you came to visit me." Through this passage, Jesus is conveying the message of taking care of the needy. This is inspiring to healthcare workers, as the main job is to take care of those in need physically and mentally. Most healthcare workers describe their purpose and desire as simply to help people, but that passion holds much more weight than most believe. Christians are also reminded in 1 Peter chapter 4 that each of you should use whatever gift you've received to serve others as faithful stewards of God's grace in its various forms. Healthcare providers and microbiologists are ultimately conveying the love of God that they have received to others by simply caring for them.